今ご覧になられているのは抗生物質の中で成長するバクテリアの様子です。普通バクテリアって抗生物質の中では成長できません。しかし突然変異が起こると抗生物質の中でも生き残れるようになるんです。さらに突然変異が続くと抗生物質の濃度の10倍、100倍、そして10日以内には1000倍以上の濃度でも生き残れるようになります。今皆さんはまさにリアルタイムで進化を目撃しているんです。
I'm off to Michigan to see the world's longest running evolution experiment. Let's go.
リチャード・ランスキー教授に会いに行きましょう。彼はなんと33年間、大学院生たちと一緒に週末も休まず実験を続けているんです。この実験室には生きた大腸菌が12個のフラスコに入っています。この大腸菌は実験室で30年間進化を続けて生き残った運のいい者たちなんです。
So there are 12 long-term lines.
1988年、共通の祖先から12の個体群に増殖し分裂しました。その後、各個体群は独立して増殖し、どのように進化していったのでしょうか。こうした長期間観察が必要な実験は1896年からアメリカのさまざまな大学で行われています。イリノイ大学ではトウモロコシを使った実験が行われていますが、1年に1世代しか育ちません。しかしバクテリアは1日に6から7世代も増えるんです。33年間でランスキー教授のフラスコの中のバクテリアは74,500世代に達しており、これは人類の進化でいうと150万年分に相当します。
What we do to start the experiment is we dilute a large population of bacteria onto a petri dish, and each individual cell makes a colony. And so then we take a little bit from one colony to start one population, a little bit from another colony to start another population, but effectively they all started from individual cells, and that's important because it means if we see the same thing happening in replicate populations, it's not because they started out with the same genetic variants and natural selection just fished out the same thing over and over. You actually had to have independent mutations that would give rise to these competitive advantages, whatever it might be, that would produce the repeatability across the 12 replicate populations.
実験室の環境はバクテリアが普段いる環境とは全然違います。すごくシンプルなんです。他の生物は存在せず、37度に保たれた中でグルコースやリン酸カリウム、クエン酸などが混ざった液体で育てられます。大腸菌の唯一の炭素源はグルコースなのですが、かなり限られているんです。
Above all else, consuming that glucose and converting it to offspring, replicating as fast as possible, has been essentially what we seem to be selecting for.
毎日フラスコの中でバクテリアは6から7回分裂し、約100倍に増加します。バクテリアの分裂回数は時間で決まると思われがちですが、実は環境の資源が影響しているんです。もしバクテリアがいる溶液の濃度が10倍だったら、個体数も10倍に増やせる可能性があるというわけです。そしてこのプロセスは33年間ほぼ毎日続けられてきました。バクテリアが培養されているフラスコの溶液 1% を同じ溶液で構成された新しいフラスコに移していきます。これでバクテリアを100倍に希釈することになります。この方法でバクテリアは1日で成長して分裂し、個体数を100倍に増やすためのスペースと資源を得られるのです。このプロセスは次の日も、その次の日も、そして週末も30年以上続けられてきました。では、新しいフラスコに移されなかった 99% のバクテリアはどうなるのでしょうか。
That's the autoclave room.
What happens in the autoclave room?
Every day, 99% of the E. coli meet their demise in this horrible room.
Is this like a bacterial crematorium?
Yeah, exactly.
もし科学者たちが毎日100倍の濃度の溶液をバクテリアに与えたとしたら、実験が手に負えなくなってしまうのです。2日目には1立方メートルの溶液が必要なのですが、13日目には地球の体積の10倍、42日目には 観測可能な宇宙全体を埋め尽くすほどバクテリアが増加してしまいます。
To me, it seems like the whole idea of genetics is for them to stay constant and for mutations to be rare.
Yes.
Yet in your experiment, it seems they're not rare.
So we estimate that in our bacteria, only about 1 out of maybe 100 or 1 out of 1,000 cells will have even a single mutation. So that's not very much. By contrast, in humans, it's estimated that each one of our offspring has perhaps 10, 20, 50 new mutations. So the bacteria are extremely conservative, but there are also billions of them in even a tiny flask.
もしも何千もの突然変異と数十億の個体数があれば、フラスコの中で毎日数百万もの新しい突然変異に出会うことになりますよね。自然選択がどんな風に作用するかによって様々な突然変異が生まれてくるのです。おそらく突然変異の半分はこの特別な環境での増殖能力には全く影響を与えません。実験室の外では役に立つ可能性があっても、実験室の中ではそうではないのです。残りの半分の中には害を与えるものもあるかもしれません。しかし、こうして毎日発生する多くの突然変異のうち、10個、100個、もしくは1000個ほどは細胞内で何かを変え、生存競争でも有利になります。そして、そういった突然変異はどんどん分裂していきます。毎日 99% は消えてしまいますが、運のいい 1% が残り、どんどん早く増殖していくバクテリアは、フラスコの中でも生き残る確率が高いのです。そして、これが幾何級数的に増えていきます。
So the mutations, are really rare when they first occur, and many of them are lost. But once they get common, if they have that competitive advantage, they'll just sweep through the population.
でも、どうやって突然変異した個体が環境に適応しているとわかるのでしょうか。ここでバクテリアの独特な特性が役に立ちます。それは長期間冷凍されていてもまた生き返ることなんです。
And so they're stored in suspended animation here. So these are the racks that contain the frozen samples of the bacteria from the various generations.
約500世代ごと、つまり人間の時間で75日ごとにランスキー教授のラボでは各個体群を冷凍保存して記録を残しています。
So our samples from over 30 years ago remain perfectly viable. And so that gives us an ability to do what I like to call time travel. We can literally compare organisms that lived at different points in time. So we can compete bacteria from generation 70,000 against their ancestor.
要するに研究者たちがバクテリアの適応度を測定する方法は今の世代のバクテリアと昔の世代のバクテリアを競わせることなんです。研究者たちは昔の世代を解凍し、今の世代が入ったフラスコに混ぜた後、培養液を入れたフラスコに注入してどちらの世代が多くなるかを見てみました。1日培養した後にまたプレートに移してどれくらい成長したかを比べるんです。どの世代がグルコースをうまく使い、より早く分裂できるかを確認します。
Well, how the heck do you tell an evolved bacterium from its ancestor? Do they wave little flags at you and say, hey, I'm the evolved guy? And of course they don't. But what we have is this color marker embedded in the experiment. So 6 of our populations on a certain kind of agar plate make red colonies, and 6 of them make white colonies. And we have one version of the ancestor that makes red colonies and one that makes white colonies. We can compete one of the red evolved populations against the white ancestor, or one of the white evolved populations against the red ancestor, and we can distinguish them.
今ご覧になられている画面には赤いコロニーが白い祖先群を超えているのがはっきりわかりますよね。助手の大学院生たちがすべてのコロニーを手作業で数えてくれています。
So what was the earliest sort of big findings from the experiment?
The first thing we found, not that there was any doubt about it, but it's one of the most direct demonstrations of Darwinian adaptation by natural selection you can imagine. Yes, they're getting to be better competitors over time. It's a common observation in other evolution experiments that evolution in a new environment gets off to a rip-roaring start and then tends to slow down over time. And so we repeated that observation, and I imagined that the long-term lines would actually sort of flatline at some point. And I actually thought about stopping the experiment, but I got wise advice from colleagues and from my wife Madeline, let's keep it going, and so I agreed to that.
教授が実験を続けてくれたことはラッキーでした。2003年に入ってバクテリアが驚くべきことを始めたのです。
One of the 12 lineages suddenly began to consume a second carbon source, citrate, which had been present in our medium throughout the experiment. It's in the medium as what's called a chelating agent to bind metals in the medium, but E. coli, going back to its original definition as a species, is incapable of that. But one day we found one of our flasks had more turbidity. I thought we probably had a contaminant in there. Some bacterium had gotten in there that could eat the citrate and therefore had raised the turbidity. We went back into the freezer and restarted evolution. We also started checking those bacteria to see whether they really were E. coli. Yep, they were E. coli. Were they really E. coli that had come from the ancestral strain? Yep. So we started doing genetics on it. It was very clear that one of our bacteria lineages had essentially, I like to say, sort of woken up one day, eaten the glucose, and unlike any of the other lineages, discovered that there was this nice lemony dessert. And they'd begun consuming that and getting a second source of carbon and energy. Zach was interested in the question of, well, why did it take so long to evolve this, and why has only one population evolved that ability? He went into the freezer and he picked bacterial individuals or clones from that lineage that eventually evolved that ability, and then he tried to evolve that ability again, starting from different points. So in a sense, it's almost like, well, it's like rewinding the tape and starting, let's go back to the minute 5 of the movie, let's go back to minute 10 of the movie, minute 20 of the movie, and see if the result changes depending on when we did it. Because this citrate phenotype, there were essentially two competing explanations for why it was so difficult to evolve. One was that it was just a really rare mutation. It wasn't like one of these just change one letter. It was something where maybe you had to flip a certain segment of DNA and you had to have exactly this breakpoint and exactly that breakpoint, and that was the only way to do it. So it was a rare event, but it could have happened at any point in time. The alternative hypothesis is that, well, what happened was a series of events that made something perfectly ordinary become possible that wasn't possible at the beginning because a mutation would only have this effect once other aspects of the organism had changed. To make a long story short, it turns out it's such a difficult trait to evolve because both of those hypotheses are true.
この実験では他にも驚くような発見がありました。例えば、時間が経つにつれてバクテリアの数が多くなるだろうという予想とは違い、数は減りましたが、サイズがどんどん大きくなっていったのです。12の系統のうち、6つは祖先よりも100倍も高い突然変異率を持っていましたが、後にその変異率を下げるという突然変異も起こりました。突然変異が多いと、他のバクテリアよりも進化が早く進みますが、あまりに多すぎると有害な突然変異が増えてしまう可能性もあるんです。
This view I had that they were flatlining turned out to be quite false. I had sort of imagined a very simple mathematical model. You can create something called a rectangular hyperbola, I guess, which has an initial high slope and then reaches an asymptote. But they're equally simple models. There's a model that also has just two parameters called a power law model that says things slow down, but it doesn't have an upper bound. It says just keep going for time immemorial, and things will just keep going faster, but at a slower and slower rate of further improvement. And it turned out that model actually fits our data better than that original model I had imagined. And not only does it fit it better, okay, you say statistics, science, fitting curves, it actually predicts the future. And that's what's really cool, because the original model, if you give it, say, just 5,000 generations worth of our fitness data and ask it to predict into the future, it says the asymptote is here. But then when we get more data, no, the bacteria are up here. They've passed that asymptote. Whereas this power law model, which says things are slowing down but never reaching an asymptote, we give it just 1/10 of our data from the past, and it projects very accurately out to 50,000 and even 60,000 generations when we last looked. It predicts sort of the future course of the evolutionary trajectory. And to me, that's kind of profound, and it's sort of changed the way I look at this experiment and even a little bit how I look at life on Earth. I mean, life on Earth doesn't stop evolving, we know that, but we think that's because they're asteroid impacts, that's because of human impacts, that's because they're viruses that are attacking their hosts, and the coevolution is causing evolution to never stop. The world is always changing, so of course evolution never stops. And that is 100% true. But what our experiment suggests is that even in the absence of environmental change, there are so many opportunities of smaller and smaller magnitude to continue to make progress that in fact, progress probably would never stop even in a constant environment. To me, it's one of the reasons to keep this experiment going. Does this model continue to predict the unfolding of the future fitness trajectory?
この動画が面白いと思ったら、高評価とチャンネル登録もよろしくお願いいたします。